What is the race for the double helix?
Great minds on both sides of the Atlantic Ocean were intent on finding the structure of DNA. Race for the Double Helix is a BBC TV production, 1986. Based on the research and personal interactions of those who worked on the structure of DNA during the 1950s. The main characters included James Watson, American, 1928, Francis Crick, British, 1916 to 2004. Rosalind Franklin, British, 1920 to 1958, Maurice Wilkins, British, 1916, Peter Pauling, Linus Pauling's son. American, 1931 to 2003, John Randall, British, 1905 to 1984, and Edwin Chargaff, French, 1905 to 2002.
Are bacterial cell walls different from plant cell walls?
Cell walls in prokaryotes, e.g., bacteria and plants define the cell's shape and give rigidity to the cell. Unlike plant cell walls, bacterial cell walls consist mainly of peptidoglycans and not cellulose. Peptidoglycans are polysaccharide chains, amino sugars, cross-linked by small peptides.
What is a wetland?
A wetland is an area that is covered by water for at least part of the year and has characteristic soils and water-tolerant plants. Examples of wetlands include swamps, marshes, bogs, and estuaries.
What are killer bees?
Africanized honeybees. The term entomologists prefer rather than killer bees are a hybrid originating in Brazil, where African honeybees were imported in 1956. The breeders, hoping to produce a bee better suited to producing more honey in the tropics, instead found that African bees soon hybridized with and mostly displaced the familiar European honeybees. Although they produce more honey, Africanized honeybees, Apis mellifera scutellata, also are more dangerous than European bees because they attack intruders in greater numbers. Since their introduction, they have been responsible for approximately 1,000 human deaths. In addition to such safety issues, concern is growing regarding the effect of possible hybridization on the U.S. beekeeping industry. In October 1990, the bees crossed the Mexican border into the United States. They reached Arizona in 1993. In 1996, 6 years after their arrival in the United States, Africanized honeybees could be found in parts of Texas, Arizona, New Mexico, Nevada, and California. Their migration northward has slowed, partially because they are a tropical insect and cannot live in colder climates, Experts have suggested two possible ways of limiting the spread of the Africanized honeybees. The first is drone flooding, a process by which large numbers of European drones are kept in areas where commercially reared European queen bees mate, thereby ensuring that only limited mating occurs between Africanized drones and European queens. The second method is frequent requeening, in which a beekeeper replaces a colony's queen with one of his or her own choosing. The beekeeper can then be assured that the queens are European and that they have already mated with European drones.
Who was Ivan Pavlov?
Ivan Pavlov, 1849–1936, was a Russian physiologist who became famous for his experiments with dogs, in which the animals performed a specific behavior upon being confronted with a certain stimulus. In these well-known investigations, minor surgery was performed on a dog so that its saliva could be measured. The dog was deprived of food, a bell was sounded, and meat powder was placed in the dog's mouth. The meat powder caused the hungry dog to salivate. This is an example of an unconditioned reflex. However, eventually, after many trials, the dog would salivate at the sound of the bell, without meat powder being offered. This is an example of a conditioned reflex or classical Pavlovian conditioning. Although he never thought much of the then fledgling science of psychology, Pavlov's work on conditioned reflexes has been far-reaching, from elementary education to adult training programs. Pavlov was awarded the Nobel Prize in Physiology or Medicine, 1904, for his study of the physiology of digestion.
What is Darwin's warm pond?
Darwin's idea of a suitable milieu for the origin of life on Earth was a little pond gently warmed by sunlight. This can be contrasted to the Oparin-Haldane hypothesis of an electrically active methane and carbon dioxide atmosphere for the origin of organic evolution.
What contributions did Overton and Langmuir make to the study of membranes?
As early as the 1890s, Charles Overton, 1865–1933, was aware that cells seemed to be enveloped by a selectively permeable layer that allowed different substances to enter and leave cells at significantly different rates. He recognized that lipid-soluble substances penetrated readily into cells, whereas water-soluble substances did not. He concluded that lipids were present on the cell surface as some sort of a coat. Irving 48 Langmuir, 1881–1957, proposed that cells were covered by a lipid monolayer. His work became the basis for further investigation into the membrane structure. Langmuir was awarded the Nobel Prize for Chemistry in 1932.
What is a genome?
A genome is the complete set of genes inherited from one's parents. Genome sizes vary from one species to another. The final number for humans is yet to be determined.
Do fungi exhibit an alternation of generations similar to plants?
Organisms with life cycles that exhibit an alternation of generations have both a haploid phase and a diploid phase. During the haploid phase, reproduction is conducted through gametes, which fuse together to form a zygote. During the diploid phase, reproduction is conducted by spores. Spores develop individually and divide through mitosis, producing the gametophytes of the next generation. Because fungi have both a haploid and diploid phase, they exhibit an alternation of generations.
What is the cell theory?
The cell theory is the concept that as all living things are made up of essential units called cells. They are the fundamental components of all life. The cell is the simplest collection of matter that can live. There are diverse forms of life existing as single-celled organisms. More complex organisms, including plants and animals, are multicellular cooperatives composed of diverse specialized cells that could not survive for long on their own. All cells come from pre-existing cells and are related by division to earlier cells that have been modified in various ways during the long evolutionary history of life on Earth. Everything an organism does occurs fundamentally at the cellular level.
What is homology?
Homology is the similarity in traits between two species that is indication of their common ancestry. For example, the general characteristics of cheetahs, lions, tigers, and house cats are whiskers, retractable claws, tooth structure, and so forth. These similarities indicate that each of these traits was inherited from a feline ancestor.
What movie was made about Dian Fossey's work with lowland gorillas?
The movie Gorillas in the Mist was released in 1988. With actress Sigourney Weaver playing the role of Dian Fossey. The movie was filmed in Rwanda and Kenya and galvanized support for the plight of the gorillas.
What is the most primitive group of animals?
Sponges, phylum Porifera, from the Latin terms porus, meaning pore, and fera, meaning bearing, represent the most primitive animals. These organisms are aggregates of specialized cells without true tissues or organs. With little differentiation and integration, and with no body symmetry. A sponge's body is perforated by holes that lead to an inner water chamber. Sponges pump water through those pores and expel it through a large opening at the top of the chamber. While water is passing through the body, nutrients are engulfed. Oxygen is absorbed, and waste is eliminated. Sponges are distinctive in possessing choanocytes, special flagellated cells whose beating drives water through the body cavity and that characterize them as suspension feeders, also known as filter feeders.
What are some economically important angiosperms?
Angiosperms produce lumber, ornamental plants, and a variety of foods.
What is a reflex?
A reflex is an involuntary response formulated in the spinal cord to a specific stimulus.
How are animals classified?
Animals belong to the kingdom Animalia. Most biologists divide the kingdom into two subkingdoms. 1. Parazoa. From the Greek para, meaning alongside, and zoa, meaning animal, and 2. Eumetazoa. From the Greek eu, meaning true, meta, meaning later, and zoa, meaning animal. The only existing animals classified as parazoa are the sponges, phylum Porifera. Sponges are very different from other animals and function much like colonial unicellular protozoa even though they are multicellular. Cells of sponges can be versatile and change form and function and are not organized into tissues and organs. They also lack symmetry. All other animals have true tissues, are symmetrical, and are classified as eumetazoa.
What is analogy?
To an evolutionist, an analogous structure is one that looks similar or has the same purpose but is definitely not the result of common inheritance. For example, bats and birds both use wings to fly. And the wings have the same general shape, thin but broad in width. However, the structures were not inherited from the same ancestors. Bats were four-legged mammals before their front limbs became modified for flight. While birds are not descended from mammals at all.
What is meant by heterozygote advantage?
Heterozygote advantage, also known as balancing selection, arises when there is a survival. Selective advantage for hybrids, heterozygotes, as compared to that of either homozygous recessive genotype or homozygous dominant genotype. Some diseases are lethal only in the homozygous state.
How long does protein synthesis take in bacteria, once mRNA is formed?
The bacterium E. coli can add an amino acid in only 0.05 second. That means that synthesizing a protein of 300 amino acids takes only 15 seconds.
How do neurons carry messages?
Imagine a long trail of dominoes running from your finger to the spinal cord in your back. When you touch a hot object, the messages this action produces in neurons is similar to the reaction that would happen if you knocked over the first domino of the set. After the first domino falls, each domino that follows it would be knocked over in turn. This process is similar to what happens between neurons. The message is carried from the fingertip all the way to the spinal cord. Neurons maintain a concentration of sodium and potassium ions, Na and Nk+, respectively. The openings present in membranes allow these ions to pass through. As the sodium and potassium gates open and then close, the electrical charge of the nerve cell changes and messages are transmitted along the neuron membrane.
What is the Kyoto Protocol?
The Kyoto Protocol was an international summit held in Kyoto, Japan, in December 1997. Its goal was for governments around the world to reach an agreement regarding emissions of carbon dioxide and other greenhouse gases. The Kyoto Protocol called for the industrialized nations to reduce national emissions over the period 2008 to 2012 to 5% below the 1990 levels. The protocol covers these greenhouse gases: carbon dioxide, methane, and nitrous oxide. Other chemicals such as hydrofluorocarbons, perfluorocarbons, and sulfur hexafluoride were to be added in subsequent years.
What are simple stains?
Simple stains highlight an entire microorganism so that cellular shapes and basic structures are visible. Simple stains commonly used include methylene blue, carbolfuchsin, crystal violet, and safranine. A stain is applied to a fixed smear for a certain amount of time and then washed off, and the slide is dried and examined.
What is unusual about Amanita mushrooms?
Some of the most poisonous mushrooms belong to the genus Amanita. Toxic species of this genus have been known by the names death angel, Amanita phalloides, and destroying angel, Amanita virosa. Ingestion of a single cap of either of these species can kill a healthy, adult human. Even ingesting a tiny bit of amatoxin, the toxin present in species of this genus, may result in liver ailments that will last the rest of a person's life.
What is the function of the skeletal system?
The skeletal system is a multifunctional system. The skeletal system provides support, allows an animal to move, and protects the internal organs and soft parts of an animal's body.
What is the current model of the plasma membrane?
The current model, frequently referred to as the fluid mosaic model, is based on work completed by Seymour J. Singer, 1924, and Garth L. Nicholson, 1943, in 1972. Their research revealed that the plasma membrane is a mosaic of integral proteins bobbing in a fluid bilayer of phospholipids. This pattern is not static because the positions of the proteins are constantly changing, moving about like icebergs in a sea of lipids. Peripheral proteins are not embedded in the lipid bilayer, but are appendages loosely bound to the membrane surface. Membrane carbohydrates on the surface function as cell markers to distinguish one cell from another. This model has been tested repeatedly and has been shown to accurately predict the properties of many kinds of cellular membranes. This structure has also been confirmed using a technique known as freeze fracture electron microscopy.
What are the uses of peat moss?
Peat moss, genus Sphagnum, grows mostly in bogs. Peat moss is favored by gardeners for its ability to increase the water-holding capacity of soils. Due to large, dead cells in the leaf-like parts, it is able to absorb 5 times as much water as cotton plants. Peat moss is also used as damp cushions by florists to keep other plants and flowers damp. Species of sphagnum also have medicinal purposes. Certain Aboriginal people use peat moss as disinfectants and, due to its absorbency, as diapers. Peat moss is acidic and is an ideal dressing for wounds. During World War I, the British used more than 1 million wound dressings made of peat moss. Native North Americans used species of the genera Neem and Bryum to treat burns. In Europe, species of the genus Dichronoesia have been used to waterproof roofs.
What does this mean?
This means that structurally, the two strands of DNA, the two sides of the ladder, run in opposite directions. This property of antiparallelity allows for formation of the hydrogen bonds between the nitrogenous bases. These bonds are critical to the integrity of the molecule.
What is unique about the water lily Victoria amazonica?
It is very big. Found only on the Amazon River, this water lily has leaves that can reach up to 6 feet 1.8 meters in diameter. The flowers of Victoria amazonica reach up to 12,180 in, 30 centimeters in height and open at dusk, but only open on 2 successive nights.
How did the navel orange originate?
Navel oranges are oranges without seeds. In the early 19th century, an orange tree in a Brazilian orchard produced seedless fruit, although the rest of the trees in the orchard produced seeded oranges. This naturally occurring mutation gave rise to what we now refer to as the navel orange. A bud from the mutant tree was grafted onto another orange tree. Branches that resulted were then grafted onto other trees, soon creating orchards of navel orange trees. Every navel orange tree is derived from pieces of the tree that first produced the mutated fruit.
What do proteins do?
In a word, everything. Proteins allow life to exist as we know it. They are the enzymes that are required for all metabolic reactions. They are also important to structures like muscles, and act as both transporters and signal receptors.
Are some forms of cancer communicable?
Yes and no. Cancer cells arise from within the body. However, certain types of cancer may actually be caused by viruses, which may be transferred from one organism to another. Currently, it is estimated that viruses may play a role in as much as 15% of human cancers worldwide.
What disease is caused by protists of the genus Trypanosoma?
Species of Trypanosoma are the cause of sleeping sickness, East Coast fever, and Chagas disease. All of these diseases are common in tropical areas. The trypanosomes that cause these diseases are transmitted by biting insects, such as the tsetse fly.
Which criteria were used to form what is referred to as the classical approach of bacterial taxonomy?
In this approach, bacteria are grouped into genera and species on the basis of the following characteristics. 1. Structural and morphological characteristics including shape, size, arrangement, capsules, flagella, endospores, and gram stain. 2. Biochemical and physiological traits such as optimum temperature and pH ranges for growth, oxygen requirements, growth factor requirements, respiration, and the fermentation of end products. Antibiotic sensitivities, and types of carbohydrates used as an energy source.
What is cell culture?
Cell culture is the cultivation of cells, outside the body, from a multicellular organism. This technique is very important to biotechnology processes because most research programs depend on the ability to grow cells outside the parent animal. Cells grown in culture usually require very special conditions, e.g., specific pH, temperature, nutrients, and growth factors. Cells can be grown in a variety of containers, ranging from a simple petri dish to large-scale cultures in roller bottles, which are bottles that are rolled gently to keep culture medium flowing over the cells.
What are the most common chromatographic techniques?
The most common chromatographic techniques are paper chromatography, gas-liquid chromatography, also called gas chromatography, thin-layer chromatography, and high-pressure, or high-performance, liquid chromatography, HPLC.
What is eugenics?
Sir Francis Galton, 1822–1911, a cousin of Charles Darwin, founded eugenics. After reading Darwin's work on natural selection, Galton thought that the human species could be improved by artificial selection, the selective breeding for desirable traits. This method is often used in domesticated animals. In Galton's plan, those with desirable traits would be encouraged to have large families, while those with undesirable traits would be kept from breeding. However, Galton's theory overlooked two important points: the importance of environmental factors on phenotypic expression, and the difficulty of removing recessive traits from the gene pool. Recessive alleles can be passed from one individual to the next as part of a heterozygous genotype, thereby escaping detection for generations. Galton's work was enthusiastically adopted in both the United States and Europe. In the United States between 1900 and 1930, eugenics gave rise to changes in federal immigration laws and the passage of state laws requiring the sterilization of genetic defectives and certain types of criminals. In Europe, eugenics became a cornerstone of the Nazi movement.
What is the scientific method?
The scientific method is the basis of scientific investigation. A scientist will pose a question and formulate a hypothesis. As a potential explanation or answer to the question. The hypothesis will be tested through a series of experiments. The results of the experiments will either prove or disprove the hypothesis. Hypotheses that are consistent with available data are conditionally accepted.
What organisms are included in the kingdom fungi?
Members of the kingdom fungi range from single-celled yeasts to Armillaria ostoi, a species that covers 2,200 acres, 890 hectares. Also included are mushrooms that are commonly consumed, the black mold that forms on stale bread, the mildew that grows on damp shower curtains, rusts, smuts, puffballs, toadstools, shelf fungi, and the death cap mushroom, Amanita phalloides. Of the bewildering variety of organisms that live on the planet Earth, perhaps the most unusual and peculiarly different from human beings are fungi. Fungi are able to rot timber, attack living plants, spoil food, and afflict humans with athlete's foot and even worse maladies. Fungi also decompose dead organisms, fallen leaves, and other organic materials. In addition, they produce antibiotics and other drugs, make bread rise, and ferment beer and wine.
Why is Archaeopteryx important?
Archaeopteryx is the first known bird. It had true feathers that provided insulation and allowed this animal to form scoops with its wings for catching prey.
Who was the first to postulate that microorganisms are the cause of disease?
Robert Koch, 1843 to 1910, was the first to identify that various microorganisms are the cause of disease. His four basic criteria of bacteriology, known as Koch's postulates, are still considered fundamental principles of bacteriology. Koch was awarded the Nobel Prize in Physiology or Medicine in 1905 for his research on tuberculosis.
How has plant classification changed over the years?
The earliest classifications of plants were based on whether the plant was considered medicinal or was shown to have other uses. Duri rustica bicato the censor, 234 to 149 BCE. Lists 125 plants and was one of the earliest catalogues of Roman plants. Gaius Plinius Secundus, 23–79, known as Pliny the Elder, wrote Historia Naturalis, which was published in the 1st century. The book was one of the earliest catalogues of significant plants in the ancient world, describing more than 1,000 plants. Plant classification became more complicated as more and more plants were discovered. One of the earliest plant taxonomists was the Italian botanist Caesalpinus, 1519–1603. In 1583 he classified more than 1,500 plants according to various attributes, including leaf formation and the presence of seeds or fruit. John Ray, 1627–1705, was the first botanist to base plant classification on the presence of multiple similarities and features. His Historia Plantarum Generalis, published between 1686 and 1704, was a detailed classification of more than 18,000 plants. The book included a distinction between monocotyledon and dicotyledon flowering plants. The French botanist J.P. de Tournefort, 1656–1708, was the first to characterize genus as a taxonomic rank that falls between the ranks of family and species. De Tournefort's classification system included 9,000 species in 700 genera. The Swedish naturalist Carolus Linnaeus, 1707–1778, published Species Plantarum in 1753. It organized plants into 24 classes based on reproductive features. The Linnaean system of binomial nomenclature remains the most widely used system for classifying plants and animals. It is considered an artificial system since it often does not reflect natural relationships. During the late 18th century, several natural systems of classification were proposed. The French botanist Antoine Laurent de Jussieu, 1686–1758, published Genera Plantarum. The tome Prodroma Systematis Naturalis Regna Vegetabilis was started in 1824 by the Swiss botanist Augustin Pérème de Candolle, 1778–1841, and completed 50 years later. Another Genera Plantarum was published between 1862 and 1883 by the English botanists George Bentham, 1800–1884, and Sir Joseph Dalton Hooker, 1817–1911. Charles Darwin's, 1809–1882, ideas on evolution began to influence systems of classification during the late 19th century. The first major phylogenetic system of plant classification was proposed around the close of the 19th century. Die Naturlichen Pflanzenfamilien, the natural plant families. One of the most complete phylogenetic systems of classification and still in use through the 20th century. Was published between 1887 to 1915 by the German botanists Adolf Engler, 1844 to 1930, and Karl Prantl, 1849 to 1893. Their system recognizes about 100,000 species of plants, organized by their presumed evolutionary sequence. Systems of classifications were also developed during the 20th century. Some works focused on groups of plants, especially flowering plants, rather than all plants. Charles Bessey, 1845 to 1915, was the first American scientist to publish a system of classification in the early 20th century. Cladistics is one of the newest approaches to classification. It is often defined as a set of concepts and methods for determining cladograms, which portray branching patterns of evolution.
What technique may be used to determine the presence and concentration of various chemicals in a solution?
Spectrophotometry is based on the principle that every different atom, molecule, or chemical bond absorbs a unique pattern of wavelengths of light. Using a spectrophotometer, scientists are able to measure the amount of light that is absorbed or transmitted by molecules in solution. This value of this measurement allows scientists to identify the chemical.
What is antisense technology?
Antisense technology involves targeting a protein at the RNA level as a means of regulating gene expression. The technology involves segments of DNA that are complementary to specific RNA targets. When the modified DNA binds to the RNA can no longer produce a protein. Antisense technology is a new technique in cancer therapy. Human trials are ongoing on small cell lung cancer and leukemia. Since cancer is a disease that can be characterized by excessive production of a particular protein, antisense technology works by interfering with the RNA that produces the protein.
How long can an orange tree produce oranges?
An average orange tree will produce fruit for 50 years, but 80 years of productivity is not uncommon. And a few trees are known to be still producing fruit after more than a century. An orange tree may attain a height of 20 feet 6.1 meters, but some trees are as much as 30 feet 9.1 meters high. Orange trees grow well in a variety of soils but prefer subtropical settings.
How many kinds of sharks are there and how many are dangerous?
The United Nations Food and Agricultural Organization lists 354 species of sharks, ranging in length from 6 in, 15 centimeters, to 49 feet 15 meters. While 35 species are known to have attacked humans at least once, only a dozen do so on a regular basis. The relatively rare great white shark, Carcharodon carcharias, is the largest predatory fish. The largest specimen accurately measured was 20 feet, 4 in, 6.2 m, long and weighed 5,000 pounds 2,270 kilograms.
What is the difference between rough and smooth endoplasmic reticulum?
Rough endoplasmic reticulum are regions rich in ribosomes that manufacture proteins. These regions appear to have a pebbly surface that is somewhat similar to sandpaper. Regions with no ribosomes are referred to as smooth endoplasmic reticulum. The smooth endoplasmic reticulum is involved in the synthesis of lipids and steroids, carbohydrate metabolism, and the inactivation and detoxification of drugs and other components that may be toxic or harmful to the cell.
What are secondary metabolites and what are some specific examples of such compounds produced by plants?
Secondary metabolites are compounds produced by plants that are important for the survival and propagation of the plants that produce them. Secondary metabolites serve as chemical signals enabling the plant to respond to environmental cues or functioning in the defense of the producer against herbivores, pathogens, or competitors. Other secondary metabolites provide protection from radiation from the sun or assist in pollen and seed dispersal. They are produced at various sites within the cell and stored primarily within the vacuole. The three major classes of secondary plant compounds are alkaloids, terpenoids, and phenolics.
What is the function of nerve tissue?
Nerve tissue serves as the communication system of an animal. Nerve tissue allows an animal to receive stimuli from its environment and to relay an appropriate response.
How does a bacterium's requirement for oxygen act as a basis for classification?
Bacteria are divided into four major groups on the basis of their response to oxygen. Aerobic bacteria grow in the presence of oxygen. Microaerophilic bacteria grow best at oxygen concentrations lower than those present in air less than 20% of oxygen. Anaerobic bacteria grow best in the absence of oxygen. Facultative anaerobes can grow in the presence or absence of oxygen, with more growth when oxygen is present.
What is a gene patent?
A gene patent is a patent issued for a specific DNA sequence of a specific organism. It is common for companies to obtain patents for genes important to medicine and agriculture.
How do animals know who is the boss in the herd?
Determining dominance can be overt or quite cryptic. It may involve a physical challenge and some kind of stylized fighting. Or it may rely on body language, like a change in the way a dog wags its tail. Sometimes animals determine dominance by cues too subtle for human observers to recognize.
Which plants have been used to create dyes?
Natural materials, including many plants, were the source of all dyes until the late 19th century. Blue dye was historically rare and was obtained from the indigo plant, Indigofera tincturatoria. Another color difficult to obtain for dye was red. The madder plant, Rubia tinctorum, was an excellent source of red dye and was used for the famous red coats of the British Army.
Is DNA always double-stranded?
At temperatures greater than 176 degrees Fahrenheit 80 degrees Celsius, eukaryotic DNA will become single-stranded. Single-stranded eukaryotic DNA does not always have the characteristic secondary helical structure. Single-stranded DNA can form a hairpin, stem, or a cross. Certain viruses have only single-stranded DNA.
Are all cell membranes alike?
Although all cell membranes have the same general structure, the membrane composition is different for each species, depending on their function. Membranes vary in the amount of protein they contain or the type of membrane receptors they contain. For example, plants that can survive frigid temperatures, such as winter wheat, are able to increase their concentration of unsaturated phospholipids during the winter to prevent membrane solidification. Another example is muscle cells, which have plasma membrane receptors for the neurotransmitter acetylcholine that tells them when to contract.
How does the use of antibiotics in animal feed promote animal growth?
Farmers introduced the use of antibiotics to animal feed more than 40 years ago. The main reason for antibiotic use was, and continues to be, to keep the animals healthy. Animals are often kept closely together in pens, a condition that promotes the spread of bacterial infections. The use of antibiotics inhibited the incidence and spread of infection among livestock. An unplanned side effect of this treatment was accelerated animal growth. Scientists believe antibiotics suppress the intestinal bacteria Clostridium perfringens, which produces toxins that may retard animal growth. After this discovery, farmers began to give their animals antibiotics to promote weight gain as well as to treat infection.
How does the nucleus of an atom differ from the nucleus of a cell?
The English word nucleus, meaning inside the shell, is derived from the Latin word for nut. The nucleus of an atom is an enclosed space containing protons and neutrons. The nucleus of a cell is a membrane-enclosed organelle that contains the genetic material of a cell.
What is phylogeny?
Phylogeny is the evolutionary history of a group of species. This history is often displayed as a phylogenetic tree, in which individual species or groups of species are listed at the end of the branches. The nexuses where branches join indicate common ancestors. Systematics is the area of biology devoted to the classification of organisms. Originally introduced by Carl Linnaeus, 1707 to 1778, who based his classification system on physical traits. Systematics now includes the similarities of DNA, RNA, and proteins across species as criteria for classification.
What are the functions of stems?
The four main functions of stems are: 1. To support leaves, 2. Produce carbohydrates, 3. Store materials such as water and starch, and 4. Transport water and solutes between roots and leaves. Stems provide the link between the water and dissolved nutrients of the soil and the leaves.
What is a centimorgan?
A centimorgan, cm, is a way of measuring the distances between genes on a chromosome. This unit was named in honor of Thomas H. Morgan, 1866 to 1945, who was one of the first to map genes onto chromosomes. Typically, a distance of a cm corresponds to a distance of about 1 million base pairs.
How do I know my enzymes are working?
Obviously, it is difficult to track the activities of the 20,000 enzymes that are required by your body. However, in the case of the enzyme amylase, you can easily check to see if it is working. Amylase is an enzyme in your saliva that starts the breakdown of complex carbohydrates into simple sugars, glucose and maltose. A plain cracker held in the mouth long enough will begin this breakdown, and you will actually begin to taste the sweetness of the enzyme products. Also, lysozyme is an enzyme present in your respiratory tract secretions and tears. It prevents invasion by bacteria, which explains why the warm, moist, open environment of our eyes manages to remain relatively infection-free during a normal day.
Which antibiotics are most effective against gram-positive bacteria? And which are most effective against gram-negative bacteria?
Penicillin and its semi-synthetic derivatives are effective against gram-positive bacteria. The aminoglycosides, such as gentamicin, neomycin, and kanamycin, are effective against gram-negative bacteria. Antibiotics that are effective against a broad range of gram-positive and gram-negative bacteria are called broad-spectrum antibiotics.
What is an allele?
An allele is an alternative form of a gene. There are usually two alleles for each gene. Although the number may vary from one trait to another. Each individual inherits one allele from the mother and one from the father. Alleles for a trait are located on corresponding loci on each homologous chromosome.
What were the first vascular plants?
The word vascular comes from the Latin word vasculum, meaning vessel or duct. It is believed that the first vascular plants were members of the division Rheniophyta, which flourished about 400 million years ago but are now extinct. Members of the extinct genus Cooksonia, named for the paleobotanist Isabel Cookson. 1893 to 1973, were the first vascular plants to be identified. These plants were only a few centimeters tall, with water-conducting cells present in their stems but without roots and leaves.
How is alcohol produced?
The type of alcohol found in alcoholic beverages is known as ethanol. Ethanol is produced by yeast through the process known as alcoholic fermentation. During the breakdown of glucose to harvest energy, cells generate ethanol as a way to recycle a molecule that is crucial to metabolism. Interestingly, yeast can only survive in media, like beer or wine, that contain less than 10% alcohol by volume. This means that beverages with an alcohol content greater than 10% have been fortified with additional alcohol or have been distilled. Distillation involves the boiling off and then the condensation of the ethanol, which evaporates at a lower temperature than water.
What is the structure of the plasma membrane?
The plasma membrane is a thin membrane that surrounds and defines the boundaries all living cells. It consists of a double layer, bilayer, of phospholipids with various proteins attached to or embedded in it.
What is the Wallace Award?
In 2004, the International Biogeography Society established the Alfred Russel Wallace Award for lifetime contributions to biogeography.
What is Chargaff's rule?
Chargaff's rule is based on the data generated by Edwin Chargaff. 1905 to 2002, in his study of the base composition of DNA from various organisms. By comparing the nitrogen base composition of various organisms, he found that in all double-stranded DNA, the amount of adenine equals the amount thymine, and amount of guanine equals the amount cytosine, A equals T, G equals C.
How do hardwoods differ from softwoods?
Hardwood and softwood are terms used commercially to distinguish woods. Hardwoods are the woods of dicots, regardless of how hard or soft they are, while softwoods are the woods of conifers. Many hardwoods come from the tropics, while almost all softwoods come from the forests of the northern temperate zone. The carotenoids, pigments in the photosynthesizing cells, which are responsible for the fall colors, are present in the leaves during the growing season. However, the colors are eclipsed by the green chlorophyll. Toward the end of summer, when the chlorophyll production ceases, the other colors of the carotenoids, such as yellow, orange, red, or purple, become visible. Listed below are the autumn leaf colors of some common trees.
Who were the founders of modern bacteriology?
German bacteriologist Robert Koch, 1843 to 1910, and French chemist Louis Pasteur, 1822 to 1895, are considered the founders of bacteriology. In 1864, Pasteur devised a way to slowly heat foods and beverages to a temperature that was high enough to kill most of the microorganisms that would cause spoilage and disease, but would not ruin or curdle the food. This process is called pasteurization. By demonstrating that tuberculosis was an infectious disease caused by a specific species of bacillus, Koch in 1882 set the groundwork for public health measures that would go on to significantly reduce the occurrences of many diseases. His laboratory procedures, methodologies for isolating microorganisms, and 4 postulates for determining Agents of disease gave medical investigators valuable insights into the control of bacterial infections.
How are animals classified based on what they eat?
Animals are classified based on whether they eat plants, other animals, or a combination of both. Animals that eat only plant matter are called herbivores. From the Latin herba, meaning green crop, and vorus, meaning devouring. Examples of herbivores are cattle, deer, and many aquatic species that eat algae. Animals that eat other animals are called carnivores. From the Latin carne, meaning flesh, and vorus, meaning devouring. Lions, sharks, snakes, and hawks are examples of carnivores. Animals that eat both plants and other animals are called omnivores. From the Latin omnes, meaning all, and vorus, meaning devouring. Humans, crows, and raccoons are examples of omnivores.
What are the steps to prepare a specimen for examination?
The three basic steps to prepare a specimen are fixation, preservation, staining, and mounting. Preservation prevents destruction or decay of the specimen as well as inhibiting microbiological growth. Different stains and dyes attach to different parts of a cell, such as the nucleus. Specimens are mounted in a medium that is also a preservative and often covered with a cover slip.
What groups of organisms have prokaryotic cells and which ones have eukaryotic cells?
All living organisms are grouped into three large groups called domains. They are bacteria, archaea, and eukarya. The domains bacteria, eubacteria or true bacteria, and archaea. Archaebacteria or ancient bacteria consist of unicellular organisms with prokaryotic cells. The domain eukarya consists of four kingdoms: Protista, fungi, plantae, and animalia. Organisms in these groups have eukaryotic cells. Eukaryon means true nucleus.
What are mitochondria?
A mitochondrion, singular, is a self-replicating, double-membraned body found in the cytoplasm of all eukaryotic cells. The outer membrane of a mitochondrion is smooth, while the inner membrane is folded into numerous layers that are called cristae. Mitochondria are the location for much of the metabolism necessary for protein synthesis and the production of both ATP and lipids.
Do humans really need cholesterol?
Cholesterol is an important component in the cell membranes of animals. Cholesterol, a lipid-based molecule, steroid, actually has two functions: 1, to help stabilize the membrane. 2, to maintain membrane flexibility as temperature changes. Normally the human body is capable of producing all the cholesterol it needs. Dietary ingestion of excess saturated fats and cholesterol is currently thought to be the source of the plaque that builds up in arteries and can cause heart attacks and strokes. Dietary cholesterol can be found in all animal sources, including shellfish. Plant phytosterols have the same function as cholesterol found in animals, but they do not affect the human body in the same way.
What is a VNTR?
VNTR refers to variable number of tandem repeats, which is a kind of molecular marker that contains sequences of DNA that have end-to-end repeats of different short DNA sequences. When the repeating unit is 2 to 4 base pairs long, it is called an STR, short tandem repeat. And when the repeating unit is 2 to 30 base pairs long, it is called a VNTR. An example of a VNTR is GAT, which is 4 tandem repeats of the sequence GGAT. In addition, VNTRs are highly variable regions, and thus there may be many alleles. Gene variations, at many loci, physical location of a gene on a chromosome. VNTRs were discovered by Alec Jeffreys, 1950. and colleagues as they were working on the basics of DNA fingerprinting.
Which scientists made significant discoveries toward the theory of photosynthesis?
The ancient Greeks and Romans believed that plants derived their food from the soil. The earliest experiment to test this hypothesis was performed by the Belgian scientist Jan-Baptiste van Helmont. 1577 to 1644, who grew a willow tree in a container of soil and fed it only water. At the end of 5 years, the weight of the willow tree had increased by 164 pounds. 74.4 kilograms, while the weight of the soil had decreased by 2 ounces, 57 gr. Van Helmont concluded the plant had received all its nourishment from the water and none from the soil. Joseph Priestley, 1733 to 1804, demonstrated that air was restored by plants. In 1771 Priestley conducted an experiment in which he placed a lighted candle in a glass container and allowed it to burn until extinguished by lack of oxygen. He then put a plant into the same chamber and allowed it to grow for a month. Repeating the candle experiment a month later, he found that the candle would now burn. Priestley's experiments showed that plants release oxygen, O2, and take in carbon dioxide, CO2, produced by combustion. The Dutch physician Jan Ingenhousz, 1730 to 1799, confirmed Priestley's ideas. emphasizing that air is restored only by green plants in the presence of sunlight. Evidence of photosynthesis's two-stage process was first presented by F. Blackman. 1866 to 1947, in 1905. Blackman had identified that both a light-dependent stage and a light-independent stage occur during photosynthesis. In 1930, C.B. Van Niel, 1923 to 1977, became the first person to propose that water, rather than carbon dioxide, was the source of the oxygen that resulted from photosynthesis. In 1937, Robert Hill, 1899 to 1991, discovered that chloroplasts are capable of producing oxygen in the absence of carbon dioxide only when the chloroplasts are illuminated and provided with an artificial electron acceptor.